Hello, and welcome back to Honors Biology. So today we're going to be talking about protein synthesis. So this actual gene expression now. That's what we're going to be talking about. So we've covered, DNA, the structure, replication, and that's great. But now the question is how do, you know, what DNA contains, like it contains the information for blue eyes or for brown hair or for how tall you're going to be or how big your ears are. How does that actually get expressed then? How does that information go from DNA into a protein? What's that process? It's actually this process here. It goes from DNA to RNA to a protein. And we call this the central dogma of biology. This is almost everything else that we talk about for the rest of the year. Almost everything else we talk about the rest of the year is based on this. This is the core to understanding when we get to Mendelian genetics and patterns of inheritance. When we talk about natural selection and evolution, it all comes down to this: how genes actually get expressed. So let's start talking about it. So the problem is that our DNA has genes, right? It has your genes for your brown eyes or for how tall you're going to be or how fast your fingernails grow. I mean, there's a ton of different genes that your body codes for and creates. But the problem is if DNA cannot leave the nucleus because it's a big molecule, right? It's packed in these chromosomes and it's tightly wound up and it can't leave, then how can the instructions then get out of the nucleus to the ribosomes? That's where the proteins are made. They're made in the ribosomes. Well, on on Thursday in class we talked about RNA, and that's the role of RNA. RNA is what kind of carries the information out of the nucleus into a ribosome. So this is just more recapping of what we did Thursday. But there's three big differences between DNA and RNA. One is the sugar, all right? So DNA has that sugar of deoxyribose, which is this one right here. It's that 5-carbon sugar of deoxyribose, whereas RNA has a sugar called ribose. So that's the difference between the sugars, is that DNA has deoxyribose, RNA has ribose, which is why we call it RNA. The next difference is the nitrogen bases. DNA, as we know, has adenine, thymine, cytosine, guanine. RNA instead has adenine, uracil, cytosine, guanine. So the thymine in DNA gets replaced with uracil. You can for uracil. And then the number of strands that make up the molecule. So DNA is double stranded. We call it like double helix, because there's two of them. They create a double helix. RNA is just one strand, so there's only one helix to the DNA. So here's your three main differences between DNA and RNA. Make sure you know these differences. All right, make sure you know what each of these differences are between DNA and RNA. Cool. Let's move on. So for RNA, before we get digging into how proteins are made, we gotta talk about protein synthesis, or the different types of RNA. There's three main types, but we're only going to be talking about two of them. One is messenger RNA, or we call it mRNA. mRNA is the one that actually carries the information from the double helix, from the DNA to the ribosome. So it's the one that actually carries the information out of the nucleus to the ribosome. That's mRNA. The second type we're going to talk about is transfer RNA and tRNA, and these are just the ones that actually have that amino acid number. We're making proteins here, so proteins are just strings of amino acids. That's what they are. So each amino acid latches on to a transfer RNA to a tRNA, and then this is where it actually comes in and like, okay, it's this amino acid, it's this amino acid, it's this amino acid. So and you'll see when we kind of piece it together, the role between mRNA and tRNA. So remember your two types. The other type is ribosomal RNA or rRNA, but we're not going to really talk about that. So remember the two ones, mRNA and the tRNA. So let's actually get into protein synthesis now. So as we said, proteins are a long series of amino acids. You got leucine, cytosine, alanine, you have methionine. There's 20, 21 different amino acids. So you have an infinite number of possibilities of proteins. Proteins are just made of different recombinations of those 20, 21 amino acids. So it's a two-step process. We gotta go from DNA to RNA, and then RNA to the protein, which is your amino acids. So step one, DNA to RNA, is called transcription, and then step two, RNA to protein, is called translation. So the transcription process, DNA to RNA, I think of it as the RNA has to be like scripted down. So you're transcribing DNA to RNA, and then the RNA is then translated into a protein. So let's look at these two steps a little bit more in depthly. So the first step is transcription. So transcription, all we're talking about there is a complementary single strand of RNA, mRNA, is copied from part of the DNA in the nucleus. So we're in the nucleus, the double helix unwinds, and then we get messenger RNA, and it forms a complementary strand. The same base pairing rules apply as what we did with DNA, but one exception though. Because we don't have thymine for RNA, we have uracil instead, A is going to pair with U. So C with G, and A is going to pair with the U. Thymine, so they're going to pair with the A, G with C, but A with U. So there's your difference. So that's how this strand is built in. So an enzyme called RNA polymerase comes in and unwinds the DNA strand. And then RNA polymerase, it reads, much like how DNA polymerase came in and read each nucleotide. It's like, okay, you're a T, I have an A for you. You're a C, I have a G for you. XYZ. RNA does a similar thing, but instead of building a DNA strand, it builds a mRNA strand. So if DNA, if this sequence is TAC, CAG, TTT, then the RNA sequence, the mRNA sequence, is going to be AU, GG, UC, AAA. All right? Cool. So then mRNA leaves and then DNA coils back up. So that's what it is. So that's the first step, is transcription. We have successfully gone from DNA to mRNA. So that's the first half of the essential dogma of biology. The second half is this translation. And before we get into how it works, we have to understand how the DNA code is now read. So it's read in a series of 3. I've been kind of grouping these up, but so far in all my examples in groups of 3, and that's because every group of 3 represents a codon, and a codon is going to tell you what amino acid to put there. So for example, up here, AUG is a codon. This series of 3 is going to tell you what amino acid is produced from this codon. AAC is a different codon. This is going to tell you what amino acid is made there. And amino acids of the 20 or 21, I don't know how many they are, roughly 20, 21. They're abbreviated by using the first 3 letters of the amino acid's name. So, because they get super long names like methionine, we just abbreviate it MET, M-E-T, and it works. MET is methionine. Look at that. I didn't know what example I had. So, like methionine is MET. LEU is abbreviated because it's leucine. Some of them get really long. Like there's phenolphthalein, but we abbreviate it PET or PHE, I think. So we can actually determine what amino acids get produced. So I know this chart looks very intimidating, but let me bring it out. This chart is also uploaded in Google Classroom, so you can actually go on into Google Classroom and use it, which I highly recommend you doing because I do ask questions in future assignments about what amino acids are produced. So make sure you're very familiar with this chart. So how you read this chart and how it works is really straightforward. So you start with your codon, your group of three, A, U, G. Fun fact, your very first codon, very first amino acid will always be AUG, and I'll explain why. So we look at the nucleotide, the base in the first position, and it's A. So we find the A on here. Okay. Then we look at the nucleotide in the second position, which is U. So then we go up here to U, and then we find what box we're in. So we're in this box here. So we have A and then U, so we're in this box, and we look at the amino acid in the third position, which is G, and we put all three together, and we get methionine. CAU, our first nucleotide is the C, second one is A, third one is U, so we pronounce histidine. UAG is a stop codon. If you notice, there's 3 stop codons. So U for the first codon, then A, then we find G, stop. So it's a really straightforward process. So I asked a question right now in the video. It should pause and ask you, what is the amino acid produced from this first guy, GCU? So pause. You answer the question. It's alanine. And then the rest of these, so GCU, GCU is only alanine. UAC is tyrosine or tyrosine. CUG is leucine again. UUA is leucine. So if you notice, there's a lot of redundancy in this code. It's redundant, right? You can get really, if it's CC, then any third letter is going to produce probably, or take leucine for example. There's 1, 2, 3, 4, 5, 6. Looks like there's 6 different combinations that ways that we can get leucine, and we'll explain why the DNA code is redundant. It's actually a really smart purpose. It's to help fight mutations that can occur. So that's how you read, and you'll get more practice with it. I promise. So this only works though for the mRNA code. So don't try applying it to tRNA or to DNA. It's not going to work. Only works for the mRNA. So let's actually now talk about translation. Now that you kind of introduced how this code is read. So translation occurs in the cytoplasm at a ribosome. So this is where then we turn that mRNA into a protein, to an actual series, a chain of amino acids. So let's get into that process. So mRNA, as it moves through the ribosome, right? So the mRNA leaves the nucleus, goes to the ribosome. When it attaches to the ribosome, the ribosome is going to read it. And it's like, okay, so your AUG. Here comes in this tRNA, this transfer RNA strand that has the correct codon, has the correct amino acid. tRNA brings in the amino acid that is the correct amino acid, and then binds, is like, okay, methionine, you're there, it binds methionine then to the peptide, and then it releases it. So we call the tRNA, we call it the anticodon because it's a complementary process. So here's your mRNA strand, which is your codon, and then their tRNA with an anticodon just matches the amino acid to it. That's all the tRNA is doing, is it's just coming in and it's bringing in the correct amino acid. That's it. But remember, the mRNA is what determines what amino acid is kind of produced or used, not tRNA. mRNA is what determines what amino acid is produced. Right. Then the last little part of this is, so protein synthesis is regulated, right? So take our cells, for example. Every cell in our bodies has the exact same copy of DNA, but they have to do different things, right? The cells in your heart have to make different proteins than the cells in your liver or the cells in your brain or the cells in your legs, right? They all have to make different proteins. So how do they do that if they all have the exact same copy? When they all just make the same proteins? No, because your body, your cells are in your DNA is really smart. What it does is, remember how I said that you'll find AUG methionine at the start of every DNA or every mRNA sequence? That's because it's the start codon. So when the mRNA strand is produced and it goes to the ribosome, the ribosome will read it until it finds the AUG. Once it finds AUG with methionine, it will be like, okay, we are now starting translation. So once it reads the AUG, then it knows the next strand or whatever comes after that is making the protein. This is the gene that it is inscribing for. And then at the very end, then how does it know to stop? Where does it know to stop making these proteins? And it's because there's three proteins here three codons that tell it to stop. We call them stop codons. And they're found at the end. They don't code for an amino acid. There's no amino acid that codes for. It just tells it to stop. All right? So I know that was a lot. I know we talked about some of it on Thursday. Hopefully this filled in the gaps and got you thinking more. If you have questions, make sure you ask them at the very end where I ask that question. And then we'll talk about it more on Monday. I'm super excited because now we can talk about genes and expression, and we can get into now mutations where what if this process doesn't work. So I'll see you all on Monday. Bye.